We will move to the second chapter, that is a biological classification. This chapter is very interesting and it gives you a lot of ideas how these organisms are arranged for our study purpose. So, in the biological classification, what they're doing is the data's what they got through this key, that means taxonomical aids and other things. They will study that, they will arrange them in their hierarchical orders on the basis of their similarities and dissimilarities. So, why we have to arrange them? The why we have to arrange them is we know that life is originated almost millions of years ago on this earth's surface. We have different organisms, different types of organisms. So in order to study them, in order to group them, and in order to understand in which locality they are growing, and in order to understand what is the relationship between this organism to other organism in a group and outside of the group, and how these organisms are evolutionarily arranged. So this is the main motto behind this biological classification. It is just whatever the life is present on our earth surface, we are arranging them on the basis which made by the human standards, the rules made by the human standards, and studying them. So the first person who started about this classification, it is none other than it is Aristotle. So Aristotle is considered as the father of science. And what he did is Aristotle, it is a little historical part. So Aristotle is divided the life into two systems. One is with the red blood and without red blood. Anemia and anemia, that means without. And that classification he found it very difficult because most of the organisms have red blood cells and very few organisms are there without the red blood or some other colored blood. So he found out it very difficult, so that a classification was not proper. And when and Aristotle started in animals to classify this, and his disciple, who is known as Theophatus, he also started to do it in plants. So Aristotle is considered as the father of biology. Aristotle is considered as the father of zoological classification, and his disciple Theophatus is considered as the father of botany or father of botanical classification, because he started some basic botanical classification work. And after this, when the human system was growing, and when Carlos Linnaeus, the father of taxonomy in 1758, when he introduced that system of binomial nomenclature and other thing, so what he did is he divided in a whatever by his understanding, he divided into two forms. One is Plantae and Animalia. So it was very simple things. So some few things he was knowing about the cellular level and other thing, nutritional level, cell wall, and response to the stimuli is short, long, many things by normal classification of plants and animals he did. But that also found not good, because there are some organism like protista and other we can't put neither in plants nor in animals. So in Ernest Haeckel in 1866, he brought the three kingdom classification, that is Plantae, Protista and Animalia. uh, and limitations, there are also limitation, there was no separation like eukaryote or prokaryote, and both unicellular multicellular organisms protista are put in one. By studying that, four kingdom classification came to exist, which was put forward by the Copeland. Also he introduced another kingdom known as monera, plantae, fungi, these things are introduced. And finally in 1969, the five kingdom classification, Now it is updated to six kingdom classification, and the five kingdom classification is considered to be not 100% accurate, but up to the level it is best. We saw it. So five kingdom classification was done by the Whitaker. So he divided on the basis of cell structure, mode of nutrition, reproduction, thallus organization, phylogenetic relationship, that is evolutionary relationship. He divided it. So the first thing was studied is monera, and monera means, that means in his five kingdom classification there were five: monera, protista, fungi, plantae, animalia. Five. So that is known as five kingdom classification in Veritas five. And now there is six kingdom classification also, and now they are proposed for seven kingdom, but we are not following, and more still the most common followed is five kingdom classification. So the kingdom Monera, and in kingdom Monera, what he found is Monera means the family of that means where bacteria are present, and these organisms, when we see about its cell structure, they are unicellular in nature, single cell. They don't have proper nucleus, a word of nucleus, and we can see them in most of the environments wherever we go. So these things, and the general structure of Moniran is like this. That means very simple. It is copied from net. That is, and very simply, when you see these Monirans, you can see that it's a very simple structure with a flagellum, and inside flagellum, and the flagellum is attached. Flagellum is used for the movement. Then some of them have a capsule. Capsule means a thick covering over the surface so that it will be resistant to these antibiotics and other chemicals. So most of the organisms, not most, all of the organisms which are having capsules, it is considered as virulent. That means they can cause disease, and they have some of them have cell wall, some of them have plasma membrane, ribosome, cytoplasm. Very simple. Pilus, pilus or fimbre, they both, and these are the characteristics of this monera. That is, monerans are unicellular in nature, and it is having a ribosome which is of 70s. DNA is naked, and there is no, and they lack cell organelle. This was studied in lower classes, and they reproduce asexually by budding or fission. Then cell wall is made up of peptidoglycan. Flagella will be present, and the mode of nutrition: autotrophic, parasitic, saprophytic. Many of them, and inside this class, the sole occupant is and the if we go behind those history, the Anton van Leeuwenhoek was the first person to see the living cell under the microscope. Then it was taken by, given to Robert Hooke, we got cell. So by the development of microscope, the study of bacteria become very prominent. So we came to know about different microorganisms and different types of bacteria, and these bacteria can be divided in many shapes and sizes according to the shape, which is cocci. That means it will be spherical in shape, and bacilli, it will be uh, rod shaped. Vibrios means kidney shaped or coma shaped. Then spirula means spiral in nature, coiled structure. Then some of them are budded. That means something like a bud. That's staphylococcus, those organism. And when we come to this morning, these are the major characteristics that we have to study from that. When we classify this monera, these monerans are again, the subkingdom is again divided into two. One is known as Archaebacteria and another one is known as Eubacteria. Archaebacteria means these bacteria are extremophiles. Extremophiles means they can live in a surrounding in extreme conditions, and on their bacteria, in order to survive them, they have some structures different on the cell wall structure, and the mode of nutrition for them is autotrophic, and the nucleotide sequence that we'll discuss later. The new bacteria, which means true bacteria, that means it is having a very rigid cell wall and made up of peptidoglycan, and it can move with the help of flagella, and sexual reproduction through pili is common, not common, but we can see them, and it pili is also help them to attach with the host. And this u bacteria can be divided into gram positive and gram negative on the basis of it's a stain that is used. And there are many new bacteria coming inside that. Then comes the cyanobacteria. Cyanobacteria means the blue green algae. It is not earlier there was also known as blue green algae. Earlier it was. Then these are photosynthetic in nature, and they have chlorophyll, carotenoid, and phycoglobulin inside them, and they're found in the aquatic region. And one of the most important thing is that these organisms can fix atmospheric nitrogen, and nostoc, anabina, spirula, these are the one of the common find cyanobacteria inside them. And some of the monerans are very useful, that means they are helpful for the nitrogen cycle nitrogen fixations. And there is one more group known as methanogens, that is important role in the sewage treatment, which is found inside the stomach of the cow. So they are also the methanogens are coming inside the archaebacteria. And apart from this, there is one more that is known as mycoplasmas. Mycus, earlier when they have found this mycoplasma in 1950s, mycus, it means a fungus shaped structures they have found. And these mycoplasmas are known as the jokers in the microbial kingdom, because they don't have a proper peptidoglycan layer on that, so they have come out of filters, and it was bacterial filters, it will escape from that. And the first report of this was came in 1898 by Knockard and Rocks. So these are the first, and they called it as PPLOs, that is pleuropneumonia like organisms. And these mycoplasmas are also called as uh, also infect to plants and animals. They can cause diseases to plants and animals. And they are very simple in nature, and they are also considered they since they are causing diseases to the plants, they are also considered as a jokers in the plant kingdom. But some says that they are only said as jokers in plant kingdom, but they cause diseases to animals also. And they are very simple. They lives in soil, and it is having a double stranded DNA inside that. It is having a plasma membrane, and the ribosomes are also present inside this mycoplasma. So these are the main things that is coming under this kingdom monera. It is very simple. That means it is classified on the basis of nutrition, its structure, reproduction, and we can see sexual reproduction in bacteria also. Most of the times we say that bacteria reproduce only asexually, but no, bacteria can reproduce by sexual reproduction, which is one called as conjugation. It is also present inside these monerans. So these are the main characteristics of monera. Thank you. If you have any doubts, please comment below.